I remember when I started to understand a little bit about how diverse the mechanisms by which long-lived species become long-lived. My first intro to that was the work of Gustavo Baja and Rael Pamplona on birds, showing very low superoxide production and low lipid peroxidizability. And it was a real eye-opener to me because, of course, what this leads to is a reduction in the natural level of expression of things downstream, specifically enzymatic antioxidants like SOD. And this, for someone like myself, who's come into the field as really a theoretician, posed this enormous challenge of how can we avoid getting misled as scientists and sent down completely wrong paths by these trade-offs, well, not so much trade-offs, but epistatic mechanistic things. Do you have any insights?
Well, I think those early days of looking at birds, the assays were actually not so easy to perform. And I remember there were 2 studies that came out almost the same year comparing pigeons and rats. And they came to completely opposite conclusions about whether they upregulated antioxidant enzymes or not. So I think the time is now to sort of revisit this stuff with the better tools that we have now, and really see. I mean, at those times, we all thought that oxidative damage was the whole story, right? And all we had to do was understand that. I think that's one of the theories that came and went. I don't think it's completely dead. I think it's a player, but it's not driving the car.
Yeah, I agree with that. I think, for some birds it was shown that they actually have very high levels of antioxidant enzymes, and they just trying to keep up and remove the damage that caused by their very active lifestyle. So again, to me, it seems that if evolutionarily something makes sense, so then biology catches up and there is a mechanism that evolves to deal with that, and that's where something we can learn from.
I think that's a very good example of how more data and new insight can actually shed a much better light. Stuff with the bats with the oxphos is a bit mad because they have the highest level of antioxidants, there's a high level of oxphos, but then it seems to drop. So maybe our assumption that it's bad is wrong. And so I think only by using our new tools, having an open mind, looking at the data and explaining why it might be different, can we get better insights. So I kind of welcome it, but we have to have open minds.
Yeah. So, I mean, one real, central message from, if we look at all 3 of your talks together, is that different species have such different ways of becoming long-lived. And of course, this, from a biomedical perspective, is just terrifying, right? Because it says, what the hell can we do to know, how to translate our findings to medical research, to what would be good in the clinic. So let's talk about that for a second. What are humans good at? How do we get to have a good longevity quotient?
Well, we are good at a few things. I would say, at DNA repair, we are definitely good. Genome maintenance, we are good at multiple things, and that goes back to Steve's talk where, humans, it's difficult to improve upon, but at the same time, when you said, there are so many different mechanisms, but that it's terrifying, I would find it exciting that there are so many different mechanisms, because some species may be good in some particular ways that we humans are not good at. For example, naked mole rat makes this abundant hyaluronic acid that we don't make, so we can benefit. Whale overexpresses KIRP, so we can upregulate our own KIRP. So in this sense, I think that provides us with this toolbox that we can use. And also another thought, that although there are many different mechanisms, But some of them are conserved, because even we talk about DNA repair, maybe not all of them would upregulate the same enzyme, but overall pathway is the same. Like there is more than one way to skin a cat, but you still get to the same result. So this is why I think we can both combine those public and private so-called strategies and kind of structure the way we look at it. So in the end, we can come up with a pretty organized picture of what makes up longevity.
Yeah, I would agree. I think that it's opportunities, that the diversity. And by the way, when I get into the field, there was a huge question, are all long-lived animals long-lived in the same way? And we've now figured that out, and they're not. But I think that's outstanding for us, because some of these mechanisms are not going to be relevant to human biology, and some of them are. For instance, having 20 copies of P53 is not going to be something that's going to translate into helping humans. But on the other hand, really active forms of SIRT6 might. So I really do think that and it's an argument for increasing, breaking open the bestiary and even looking at more of these exceptional long-lived species.
I think, again, I completely welcome it. When you ask what do humans do to make them the most long-lived, we are the ultimate tool makers. So we find solutions to our problems and we make the tools to fix it. I think that does definitely help us. Antibiotics, all of our potential interventions and so forth. But again, I see this as huge potential. As an evolution biologist, I think what we'll find, well, actually DNA repair is the thing that seems to be upregulated in the majority of them. Maybe they're all doing it in slightly different ways, but that should be our target. And I think that the more information we have, as Eric said, sequence all these genomes, find all of the noise, and then pull things out and find that one single pathway to rule them all, if it's possible. But that's a typical evolution biologist approach to it all.
Well, thank you, Emma, for bringing the brain. We kind of missed that.
Let's talk about the immune system for a moment. So, of course, we're all aware that the immune system is extraordinarily pervasive in controlling how we live as long as we do and so on. One thing, Vera, that you published a few years ago, I don't think you mentioned it today, was that naked mole rats don't have thymic involution, basically, right? So one thing that I'm thinking about this from a biomedical perspective. In the past couple of hundred years, it seems like the immune systems of the human population have deteriorated, perhaps because they could, right? Because we've got antibiotics, so women with weaker immune systems are probably more fertile anyway, and so it's been rather rapid. So, I mean, how do we take this into account in determining how to do the most relevant, the most biomedically relevant experiments in the laboratory? Can we say that we should be working in animals that have an impaired immune system, like the SPF mice, for example? What are the pros and cons?
So, I mean, I'll start off. I think that we definitely don't want to live in too clean an environment. I think that's quite obvious. We don't want to become those mice, that die the next time that we encounter something that we haven't done before. So I do think we need to think about the pathogen stew that we live in. I mean, people used to say to me, well, humans are really lab mice. They really live in a lab. No, they don't. We live in a variable environment. We live in this pathogen stew. We don't eat the good food. Of course, I'm not sure that we've optimized mouse food for that matter. But I think it's an important consideration that we don't want to live too clean.
Well, maybe I'll try to challenge this a little bit, because I think there are two ways to go about it, really. So I would agree with Steve that, yes, I mean, the way we live right now, there are pathogens, there are new ones that are coming and having impact on society. But if we try to imagine sort of future world, because many pathogens, they are taxing our immune system. From transposons to endogenous viruses to exogenous viruses. They don't really, I mean, they train our immune system and they make us better prepared, but if, let's say, we could imagine that we could just get rid of all of them and become SPF mice, that in itself, maybe it's going to be good for longevity. So I'm not a strong believer one way or another. I just think that there are two strategies to pursue, perhaps, and there may be pros and cons in each one.
But I actually will take a different view than Vera. If you think about it, I mean, scary working with bats, working potential zoonosis. I mean, every year, potentially, there's 1 billion times the viruses are trying to hop just into humans from outside the world. So we are going to be faced with a future pandemic no matter what. So we got to make sure that immune system is nicely primed. They've argued that people, again, with the Spanish flu, why was it the young people that only died? They'd argued that the older population had actually been pre-exposed, and that's why it wiped out all the youth. Imagine if COVID was wiping out our children and our babies. Things would have been a bit different. So I think that we really need to prep. You need to have dirty houses, at least that's my excuse anyway. But we again need to look to species that evolved ways. So we can tweak our immune system with drugs. It's easy to drug the immune system. We need to think about doing this, getting that balance right between enough viral control and then enough control of our own immune system, potentially through drugs if you want, to be able to achieve both. And then you just tweak that when you need. If you've got a huge, horrible infection, you need more of one or more of the other.
Excellent. So I have succeeded in the main role of a moderator in finding a question where there's a degree of disparity of opinion. All right, so here's a question, I guess it's starting with Steve, really, because it starts from an experiment that Steve did some time back. I remember maybe a decade ago you were talking kind of only in the corridors at the GSA and so on about an experiment that you did when you captured mice in your barn and you bred them for only a couple of generations to see how they would behave, how differently from the, what's Rich Miller's term, mouse-like creatures that we know. And you did a CR experiment and it basically got no benefit. So I was thinking about that again, and in relation especially to what you were saying today about wildlings. Is that like better? Does it have the same problem that the wild mice have where they bite you and so you can't use grad students to do the experiments?
Yeah, so that experiment turned out to have a complex result. The result was that there was no effect. So I did the standard 40% calorie restriction. It had no effect on median longevity whatsoever. So I was all ready to write the paper that said that wild mice do not respond to, but then the last half dozen restricted animals just wouldn't die. And they just kept living and living and living. So it turned out that all the longest lived ones was a subset. Now these were wild animals, so they were genetically heterogeneous. So there were two possible interpretations, three. One, that caloric restriction simply doesn't work in wild mice. Two, that I had over-restricted them because wild mice are not selected to be gluttonous like laboratory mice. So they eat about 20% less. So it could have been that 40% was too much restriction. The third one was that the response to calorie restriction has a genetic component, and that it was good for some and bad for others and neutral for others. And I think the follow-up work that's been done both in yeast and in other mice suggests that that latter is probably the most likely explanation. And I think, you know, that humans at large, if we could do calorie restriction, which there's now enough evidence that shows humans at scale simply cannot do it, that it would be good for some people and bad for some people and not make a difference for some people.
Yeah, okay, so coming to the other part of the question, the reasons why so few people are doing experiments on wild mice, namely that they bite you, right? Is there a way around that? Like, I was wondering whether these wildlings are like perhaps kind of best of both worlds in that way.
Yeah, I don't think you have to work with wild mice. I think what you can do is you can make lab mice more like wild mice. And I actually, you know, when Pedro asked the question about, well, wouldn't this be very expensive to fiddle? I think this could be very cheap. The way I would do the last preclinical trial before, I would just rent a small section of all these pet food pet stores, and I would say, I want to have a little booth back here with my pet store mice, and I want to do a little experiment here. And I bet that would be quite cheap. And now you're having things that are living in a very human-like environment. And so I think there are ways that you don't have to. I like wild mice because I have some respect for something that's this big that will grab a hold of me and not let go, which I don't have for laboratory mice. But I don't think you need to really work with wild mice. I mean, the other thing is you can't do a lot of the frailty measures that we typically do. We try to do roto-rod with our wild mice, and they just leap off, and they don't care about falling, from that podium to the floor. They wouldn't give a second thought to it. They just jump off.
Well, that's been our experience in wild mice. No, they didn't bite us. They will open the cage and they fly out. So only those that stay, they give rise to lab mice. These are mice that were stupid enough not to escape. But I think your point is well taken. Yeah, we need to diversify laboratory animals. Mice is extremely convenient, of course, but yeah, their idiosyncrasies are maybe not always in the direction that is very useful for us. Well, naked mole rats can be maintained relatively easily in the laboratory, but even naked mole rats don't answer every question, and you don't want to do genetics on them and wait 40 years to see if something extends lifespan.
Right.
So I think there is a proper use for any kind of creature. We just have to utilize the entire bestiary.
Yeah, absolutely. And I must say, if we had a simple answer to that question, to how we could make it more practical to use a wider variety of species in a large number of labs, then things would be very different. Okay, so I think I'm going to end with perhaps a slightly provocative question. So, as you all know, I've dedicated my career to the focus on damage repair, on actually trying to rejuvenate animals that have already accumulated a bunch of damage of whatever sort. And I'm wondering, in the context of this session, how that really works out. Like, can we be so idealistic as to say that damage repair perhaps doesn't suffer so much from this diversity of mechanisms that we see across the tree of life, or even the mammalian tree of life, such that, we're different long-lived species protect against the accumulation of damage in different ways, but it's still the same kind of damage. So if we're trying to repair it, then we might be able to actually rely better on our studies of other species than we can when we look at slowing down the creation of damage.
Well, I guess I think there's just as much diversity in damage repair. You can get rid of damaged parts and make new one, or you can repair the existing damage. And my guess is there's going to turn out to be just as much diversity in that, and we'll be able to learn from that. And I think we're going to learn a lot from, the IPS cells is changing everything. The development of organoid cultures, I think, is going to change everything. It's going to allow us to do a lot of stuff in a dish that now we have to go out and catch the animals and bring them in. So I do think there's just as much diversity in repair mechanisms, and we're going to learn a lot from understanding the way that the bats repair things versus the naked mole rats versus the bowhead whale, which, by the way, Vera, I don't know if you've read my book yet, but I think those are big underestimates of the age of bowhead whales.
Yeah, thank you, Steve. For DNA damage repair, I think this is extremely promising. It comes through comparative studies, so it is really important. Now, whether the same mechanism or different, well, I would agree that it's probably a combination. For example, we see SIR2 and 6 to be active across multiple species, but then we see some unique things. Well, I mentioned KIRP. I can mention something unique for humans, for example, and that came to us as a surprise because there is this dimer Q70, Q80 that binds to DNA break, and everybody working in DNA repair field knows about them. So we found that in humans we have maybe 10 times more of this, but all other mammals have less. So that may be our ape-specific adaptation again, like we are really good at making Q70, Q80. But then, maybe we can benefit from other aspects of improvement. And also in particular, we've been comparing different pathways of DNA repair, and double-strand break repair always comes up as the most critical, which may be because this is one pathway that also linked to epigenetic stability, because that can rearrange like the whole chunks of chromosomes. So I think this is, to me, is extremely exciting, and that may be taking us to really the underlying mechanism of age-related changes. So I think we should just keep working.
Yeah, I'm with you. Absolutely have to keep working. But maybe something else to think about. Is it DNA repair or DNA damage, not care? And so potentially you see that in these long-lived species, or even in supercenturions, they're highly, highly inflamed, but they're not showing all the effects that you should because they're able to counter it. So maybe it's a different way of dealing with either you repair it or you don't care about it, or you remove it, or all the above. And I think that when we're looking at all these different species and we look at it from those different angles and see how does it actually work to achieve the still alive and healthy, that's kind of what we're looking for. But to answer that question again about with these mice and do the long-lived ones, I'm going to push for field biology. We're moving ahead leaps and bounds in the information that you can actually collect. You can put these little things. You can use fantastic new technologies. And field biology is cheap. And the information that you can get from wild populations, but you got to fund the ecologists. That's hard to get money. Governments don't seem to want to fund natural science really anymore. But you get so much information for so little that we can feed into these studies. So again, it's another way. Rather than just have mice in a pet store, imagine if you could see mice racing around the wilds of Dublin. We have a lot of mice, more rats.
Yeah, and I think you should say that field biology is fun. That's the most critical thing. That's the one thing I really miss is a lot, of field biology.
You mean like the mud in Venezuela?
The mud, the mosquitoes, the malaria. It sounds a lot worse in retrospect than it was.
That's what brought us to this field, right? And then you start to appreciate molecular part. Although I must say, I love field biology, but my problem with field biology is that, these people don't want to kill their subjects. And sometimes to understand mechanisms, you need to bring them back to the lab and take them apart.
This is why we need to have a bit of both. So you have to have the non-invasive samples. We can sequence genomes from a single cell. So now if we can optimize the field technology by all of the work that's been done in the lab, we're going to have both. Because Vera, you're completely right. Like, how are you going to make an antibody in 20 microliters of blood? You can't. You're not going to be able to. So we have to bring it both together. But this is where all our great minds come up. Humans are great at inventing solutions. So I think we can do it all.
Fantastic. Wonderful session, wonderful speakers. So we're running only 15 minutes late. I must say, I think we've done quite well to keep it to only that. So please come back in 10 minutes by 11:15. Thank you.
Thank you, guys.